Hello, in this video we will understand about the group A Streptococcus, for short gas, the Sterpt A throat and immune response as well as the vaccine challenge. We will learn a little bit about that. So basically it is the infected person, the throat problem will be more major and the mild tonsillitis or severe tonsillitis can be happen. So this person is infected the gas, the for short the group A streptococcus gas. Basically it is normal and abnormal condition. In this way, the mild tonsillitis is not any problem and it can be reversed, but the severe tonsillitis is the problem. And in this way, the severe tonsillitis, the streptococcus can be moved into the blood circulatory system, will lead to life-threatening challenge. So this is the swollen tonsil, is known as the tonsillitis, and the whitish spot will occur. In this way, this is the throat redness and the pharyngitis and the laryngitis. Here is the pharyngitis, and this is the whitish spot, the tonsillitis, severe tonsillitis. So, but it is the just understand for you. But remember, we will understand about here is the laryngitis, the swollen of the larynx. So this is the diagram I am again drawing for understanding. The diagnosis, the swabbing for checking and under the microscope we can identify, but it is difficult in the early stage of the infection. Streptococcus obligate human pathogen, remember. So for short, in treatment is antibiotics in the current time, like penicillin. Anyhow, let's begin to understand the structure of the streptococcus, the Sterpt A, the gas, the group A streptococcus. So the variety of the proteins unable to vaccines synthesis. So that is why let's begin to understand. This is the basically you can understand in this diagram. This is shows the gas. Inside is the genetic material and outside is the varieties of protein like serum opacity factors which that inhibit the other factors, and on the other hand the interleukin 8 for example will inhibit and break down like of C5a complement system, and in this way the streptococcal C5a peptidase enzyme used for it to break down, while the streptococcal pyrogenic exotoxin A will release. And in this way here is you can see when the interleukin 8 will not produce, so the neutrophil not activate and recruit not more. So in this way the neutrophil will be dysfunction for this infection. That is why it can be progress the disease further on if the antibiotics in the early time will not provide. Anyhow, the neutrophil, the inflammatory immune cells, will not respond due to the streptococcal pyrogenic exotoxin A, and it will affect the neutrophil to kill that also. And on the other hand, the SPYCEP, the streptococcal pyogenes cell enveloped protease enzyme, which that will use to induce the inflammation more, and the protease enzyme used to break down of the protein, and in this way for example the interleukin 8 cleaves by this function, remember, and the M protein is used for the it will inhibit the function of the neutrophil for it, and the fibronectin binding protein also play important role with the SFP1 will lead to inhibit the immune function, and that is why. And the chemokines degradation will occur and the phagocytosis inhibition. So in this way, the chemokines degradation and the phagocytosis will be inhibit and the modulate the PMP function and the impair the neutrophil. So the impair of the neutrophil occur in this manner in the basically in the extracellular trap. So on the other hand, here is also the arginine deaminase enzyme used to produce the group A carbohydrates, which that is used for the protection level increase. So remember, let's begin to understand the adhesion colonization. So this is the extracellular matrix and the host immune system inside. For example, suppose this is the throat lumen. So this is the gas, and it trigger the FBAA and the SFBX and SCL1 and 2, which that will lead to bind the LTA. LTA will bind, will lead to trigger also the MP and the pilus and the SFBL, which that will lead to encapsulated the gas. It means the microcolonies will be occur inside of the or outside in the endothelial cell. So this is the adhesion and colonization. Adhesion mean it will remain outside and grow, while this is the SLA capital FBP54 protein is produced will lead to SAN binding SAN with the SAN and will lead to get H and STH will lead to trigger by this function, and in this way inside is the resident macrophage neutrophil and the circulating monocytes. Remember that things, and let's begin to understand how the immune system, the innate immune cells will inhibit by this function when invade this gas. After invasion, it can be hide also between the endothelial cell, but when move inside, the dissemination will occur and infect the cell. Infected cell produce the inflammatory cytokines and inflammation increase. While on the other hand, the macrophage will the gas will escape from the macrophage by the varieties of protein and the enzyme. So that is why, and the monocyte will be inflamed by the infection because the macrophage will not work and produce more interleukin 6, 10, and tumor necrotic factor alpha. And in this way, the escape from neutrophil extracellular trap from the extracellular trap. Anyhow, let's begin to understand the vaccination. Why vaccination is useless and it is difficult and it is a challenge for the past decade. And it is difficult due to recombination, the frequently increasing gene exchange, and SNP genotyping, single nucleotide polymorphism. The single nucleotide polymorphism is the problem, and the subsequent protein sequence, the variation, and no single protein that has been exploited for vaccine development. So the so far is 100% conserved throughout all gas isolates, and in this way, this is basically can cause the disease progression and the unable to synthesize the vaccine due to the varieties of the protein. Thanks for watching. If you like this video, please make sure to subscribe, like, and share, and give the comment below for understanding better if you have any confusion. So see you in the next video about the Sterpt A Throat.